Okay, children and grandchildren.
No, but that's the whole thing. You've got literature from particular for literature, which actually sort of film but the interest that people have in science fiction always has this combination of science and
Okay, I think you'll find this is actually more of a scientific paper than a literary paper. That just demonstrates its absolute arrogance. So, this is When Genes Go Walkabout. Is it possible for science fiction to directly influence a scientific revolution? We certainly saw instances in the space program when Arthur C. Clarke and Robert Heinlein and many others wrote fiction intended to educate, inspire, and possibly influence engineers and the general public. Astronomy and physics have long been playgrounds for science fiction, but biology has tended to play a minor supplementary role. Science fiction writers, by and large, seem to prefer spaceships and lasers to petri dishes and proteins. And biologists tend not to be strong readers of science fiction, perhaps because of the paucity of biological SF and a perceived lack of connectedness or conviction. I've never found this state of affairs to be very healthy. For the last 2 decades, biology has been the most rapidly changing and inarguably the most important of all the sciences in terms of its potential impact on our daily lives. Yet with significant exceptions, many of them mentioned by Joan, and of course Joan herself here, science fiction readers and writers have lagged in their efforts in describing modern biology, and in particular modern genetics and molecular biology. As a culture, science fiction readers tend to be remarkably squeamish about their inner workings. They prefer the clean precision of mathematical certainties and digital brains to the squishy complexities of biology and mortality. Science fiction writers like to be provocative. That's our role. What we write is far from authoritative or final, but science fiction works best when it stimulates debate. And that was my direct goal when I wrote both Darwin's Radio and Darwin's Children. I am an interested amateur, an English major with no degrees in science, and I am living proof that you don't have to be a scientist to enjoy deep exploration of the sciences. A revolution is certainly underway in how we think about the biggest issues in biology, genetics and evolution. The two are closely tied, and viruses, long regarded solely as agents of disease, seem to play a major role. For decades now, I've been skeptical about aspects of the standard theory of evolution, the neo-Darwinian modern synthesis. But without any useful alternative, and since I'm a writer and not a scientist, and so my credentials are suspect, I have pretty much kept out of the debate. Nevertheless, I have lots of time to read. My writing gives me both the responsibility and the freedom to do that, and to research thoroughly and get my facts straight. And over 10 years ago, I began to realize that many scientists were discovering key missing pieces of the evolutionary puzzle. Darwin had left open the problem of what initiated variation in a species. Later scientists had closed that door and locked it. It was time to open the door again. Collecting facts from many sources, I tried to assemble the outline of a modern appendix to Darwin, using ideas derived from disciplines not available in Darwin's time, theories of networks, software design, information transfer, and knowledge and social communication. Lots of communication. My primary inspiration and model was variation in bacteria. Bacteria initiate mutations in individuals and even in populations through gene transfer, the swapping of DNA by plasmids and viruses. Another inspiration was the hypothesis of punctuated equilibrium, popularized by Stephen Jay Gould and Niles Eldredge. In the fossil record, and for that matter, in everyday life, what is commonly observed are long periods of evolutionary stability or equilibrium, punctuated by sudden change over a short span of time, at least geologically speaking, 10,000 years or less. And the changes seem to occur across populations. Gradualism, the slow and steady accumulation of defining mutations, a cornerstone of the modern synthesis, does not easily accommodate long periods of apparent stability, much less rapid change in entire populations. If punctuated equilibrium is a real phenomenon, then it means that evolutionary change can be put on hold. How is that done? How is the allegedly steady flow of mutations somehow delayed to be released all at once? I was fascinated by the possibility that potential evolutionary change could be stored up. Where would it be kept? Is there some kind of genetic library where hypothetical change is processed, waiting for the right moment to be expressed? Does this imply not only storage, but a kind of sorting, a critical editing function within our DNA, perhaps based on some unknown genetic syntax and morphology? If so, then what triggers the change? Most often, it appears that the trigger is either environmental challenge or opportunity. Niches go away, new niches open up. Food and energy become scarce, new sources of food and energy become available. Lacking challenge or change, evolution tends to go to sleep, perhaps to dream, and sometimes to rumple the covers, but not to get out of bed and go for coffee. Because bacteria live through many generations in a very short period of time, their periods of apparent stability are not millennia, but years or months or even days. Resistance to hold it a second here. The most familiar mutational phenomena in bacteria, resistance to antibiotics, can happen pretty quickly. Bacteria frequently exchange plasmids that carry genes that counteract the effects of antibiotics. Bacteria can also absorb and incorporate raw fragments of DNA and RNA not packaged in neat little chromosomes. The members of the population not only sample the environment, but exchange formulas, much as our grandmothers might swap recipes for soup and bread and cookies. How these recipes initially evolved can in many instances be attributed to random mutation or to the fortuitous churning of gene fragments acting through the filter of natural selection. Bacteria do roll the dice, but recent research indicates that they roll the dice more often when they're under stress. That is, when mutations will be advantageous. Interestingly, they also appear to roll the dice predominantly in those genetic regions where mutations will do them the most good. Bacteria, it seems, have learned how to change more effectively, more efficiently. Once these bacterial capabilities evolve, they spread rapidly. However, they spread only when a need arises. Again, natural selection. No advantage, no proliferation. No challenge, no change. But gene swapping is crucial. And it appears that bacteria accept these recipes not just through random action, but through a complicated process of decision-making. Bacterial populations are learning and sharing. In short, bacteria are capable of meta-evolution, self-directed change in response to environmental challenges. Because of extensive gene transfer, establishing a strict evolutionary tree of bacterial types has become difficult, though likely not impossible. We're just going to have to be clever, like detectives solving crimes in a town where everyone is a thief. Perhaps the most intriguing method of gene swapping in bacteria is the bacterial phage, or bacterial virus. Bacterial phages for short, can either kill large numbers of host bacteria reproducing rapidly, or lie dormant in the bacterial chromosome until the time is right for expression and release. Lytic phages almost invariably kill their hosts, but these latter types, known as lysogenic phages, can actually transport useful genes between hosts, and not just randomly, but in a targeted fashion. In fact, bacterial pathogens frequently rely on lysogenic phages to spread toxin genes throughout a population. Cholera populations become pathogenic in this fashion. In outbreaks of E. coli that cause illness in humans, lysogenic phages have transported genes from Shigella, a related bacterial type, conferring the ability to produce Shiga toxin, a potent poison. Thus, what at first glance looks like disease, viral infection, is also an essential method of communication, FedEx for genes. When genes go walkabout, bacteria can adapt rapidly to new opportunities. In the case of bacterial pathogens, they can rapidly exploit a potential marketplace of naive hosts. In a way, decisions are made, quorums are reached, genes are swapped, and behaviors changed. What lies behind the transfer of bacterial genes? Again, environmental challenges and opportunities. While some gene exchange may be random, bacterial populations overall appear to practice functions similar to education, regimentation, and even the execution of uncooperative members. When forming bacterial colonies, many bacteria, often of different types, group together and exchange genes and chemical signals to produce an organized response to environmental change. Often this response is the creation of a biofilm, a slimy polysaccharide construct complete with structured habitats, fluid pathways, and barriers that discourage predators. Biofilms can even provide added protection against antibiotics. Bacteria that do not go along with this regimen can be forced to die, either by being compelled to commit suicide or by being subjected to other destructive measures. If you don't get the picture, you break down and become nutrients for those bacterial brothers who do, thus focusing and strengthening the colony. A number of bacteriologists have embraced the notion that bacteria can behave like multicellular organisms. Bacteria cooperate for mutual advantage. Today in the dentist's office, what used to be called plaque is now commonly referred to as a biofilm. They're the same thing. Bacterial cities built on your teeth. In 1996, I proposed to my publishers a novel about the coming changes in biology and evolutionary theory. The novel would describe an evolutionary event happening in real time, the formation of a new subspecies of human being. What I needed, I thought, was some analog to what happens in bacteria. And so I would have to invent ancient viruses lying dormant in our genome, suddenly reactivated to ferry genes and genetic instructions between humans. To my surprise, I quickly discovered I did not have to invent anything. Human endogenous retroviruses are real, and many of them have been in our DNA for tens of millions of years. Even more interesting, some have a close relationship to the virus that causes AIDS, HIV. The acronym HERV, Human Endogenous Retrovirus, became my mantra. In 1997 and 98, I searched the literature and the internet for more articles about these ancient curiosities and located a few pieces here and there. Occasional mention in monographs, longer discussions, and a few very specialized texts. I was especially appreciative of the treatment afforded to HERV in the Cold Spring Harbor text Retroviruses, edited by Drs. Coffin, Varmus, and Hughes. But to my surprise, the sources were few, and there was no information about HERV targeted to the general layman. As a fiction writer, however, I was in heaven. Ancient viruses in our genes, and hardly anyone had heard of them. If I had had any sense, I would have used that for what it seemed at face value, a ticking time bomb waiting to go off and destroy us all. But I had different ideas. I asked, what do HERV do for us? Why do we allow them to stay in our genome? In fact, even in 1983, when I was preparing my novel Blood Music, I asked myself, what do viruses do for us? Why do we allow them to infect us? I suspected they were part of a scheme involving computational DNA, but could not fit them in. Not just then. HIV was just coming into the public consciousness, and retroviruses were still controversial, believe it or not. I learned that HERV expressed in significant numbers in pregnant women, producing defective viral particles apparently incapable of passing to another human host. So what were they? Useless hangers-on? Genetic garbage? Instinctively, I could not believe that. I've always been skeptical of the idea of junk DNA, and certainly skeptical of the idea that the non-coding portions of DNA are deserts of slovenly and selfish disuse. HERV seemed to be something weird, something wonderful and counterintuitive, and they were connected with HIV somehow, a species-crossing retrovirus that had become one of the major health scourges on the planet. I couldn't understand the lack of papers and other source material on HERV. Why weren't they being investigated by every living biologist? In my rapidly growing novel, I wrote of Kay Lang, a scientist who charts the possible emergence of an HERV capable of producing virions, particles that can infect other humans. To her shock, the HERV she studies is connected by investigators at the CDC with a startling new phenomena, the apparent mutation and death of infants. The infectious HERV is named Shiva, but Shiva turns out to be far more than a disease. It's a signal prompting the expression of a new phenotype, a fresh take on humanity, a signal on Darwin's radio. In 1999, the novel was published. To my gratified surprise, it was reviewed in Nature and other science journals. Within a very few months, news items about HERV became far more common. New scientific papers reported that ERV-related genes could help human embryos implant in the womb, something that has recently been given substantial evidence, or substantial credence. And on the web, I encountered the fascinating papers of Dr. Louis P. Villarreal. I felt as if I had spotted a big wave early and jumped on board just in time. While there is certainly no proof that retroviruses do everything I accuse them of in Darwin's radio, we now know of selected instances of infectious endogenous retroviruses, though not in humans. Parasitic wasps produce viruses out of their genomes to infect the caterpillars that will host their offspring and subdue them. A direct instance of a process that once again, I thought it might be pure speculation when I used it as a major theoretical plot point in Darwin's Children. So after 5 years and 2 years respectively, both of my speculative novels hold up fairly well, and I've learned a major lesson. I can't invent anything weirder than what's already in biology. According to Wired magazine, I still have street cred. Biotech CEOs and scientists still maintain that my biology is sound. They all pick out different things that aren't sound, but they don't agree with each other as to which things they are. If my speculative biology is sound, then the only conclusion I can draw is that the biotech revolution will very soon hit contemporary evolutionary theory square across the chops. And for me, scientifically speaking, the parallel of HERV with lysogenic phages is still startling. So back to the real world of evolution and genetics. The picture we see now in genetics is complex. Variation can occur in a number of ways. DNA sequence is not fate. I don't know how many times I have to tell that to people who consider themselves experts in the field. If they say that, they don't know their stuff. Far from it. The same sequence can yield many different products. Complexes of genes lie behind most discernible traits. Genes can be turned on and off at need. Non-coding DNA is becoming extremely important to understanding how genes do their work. As well, mutations are not reliable indicators of irreversible change. In many instances, mutations are self-directed responses to the environment, and changes can be reversed and then reenacted at a later time, and even passed on as reversible traits to offspring. Even such neo-Darwinian no-nos as the multiple reappearances of wings and stick insects points toward the existence of a genetic syntax, a phylogenetic toolbox, rather than just random mutation. Wings are in the design scheme, the bow plan. When insects need them, they can be pulled from the toolbox and implemented once again. We certainly don't have to throw out Mr. Darwin. Natural selection stays intact. Random variation is not entirely excised, but the neo-Darwinian dogma of random mutation as a cause of all variation, without exception, has been proven wrong. Like genetics, evolution is not just one process, but a collaboration of many processes and techniques. And evolution is not entirely blind. Nor must evolution be directed by some outside and supernatural intelligence to generate the diversity and complexity we see. Astonishing creativity we're discovering can be explained by wonderfully complicated internal processes. These newer views of evolution involve learning and teamwork. Evolution is in large part about communication, comparing notes and swapping recipes, as it were. It appears that life has a creative memory and knows when and how to use it. Let's take a look at what the scientists have discovered thus far. Viruses can and do ferry useful genes between organisms. Viruses can also act as site-specific regulators of genetic expression. Within a cell, transposable elements, jumping genes similar in some respects to endogenous retroviruses, can also be targeted to specific sites and can regulate specific genes. Both viruses and transposable elements can be activated by stress-related chemistry, either in their capacity as selfish pathogens, a stressed organism may be a weakened organism, or as beneficial regulators of gene expression, a stressed organism may need to change its nature and behavior. Viral transmission occurs not just laterally from host to host, often during sex, but vertically through inherited mobile elements and endogenous retroviruses. Chemical signals between organisms can also change genetic expression. As well, changes in the environment can lead to modification of genetic expression in both the individual and in later generations of offspring. These changes may be epigenetic. Factors governing which genes are to be expressed in an organism can be passed on from parent to offspring, but also genetic in the sequence and character of genes. Our immune system functions as a kind of personal radar, sampling the environment and providing information that allows us to adjust our immune response, and possibly other functions as well. These pathways and methods of regulation and control point toward a massive natural network capable of exchanging information, not just genes themselves, but how genes should be expressed and when. Each gene becomes a node in a genomic network that solves problems on the cellular level. Cells talk to each other through chemistry and gene transfer, and through sexual recombination, pheromonal interaction, and viruses, multicellular organisms communicate with each other and thus become nodes in a species-wide network. On the next level, through predation and parasitism, as well as through cross-species exchange of genes, an ecosystem becomes a network in its own right, an interlinking of species both cooperating and competing, often at the same time. Neural networks from beehives to brains solve problems through the exchange and selective cancellation and modification of signals. Species and organisms and ecosystems live and die like signals in a network. Death, the acts of natural selection, is in itself a signal, a stop code, if you will. Networks of signals exist in all of nature, from top to bottom, from gene exchange to the kinds of written and verbal communication we see at this event. Changes in genes can affect behavior. Sometimes even speeches can affect behavior. Evolution is all about competition and cooperation and communication. Traditional theories of evolution emphasize the competitive aspects and de-emphasize or ignore the cooperative aspect. But developments in genetics and molecular biology render this emphasis implausible. Genes go walkabout far too often. We are just beginning to understand the marvelous processes by which organisms vary and produce the diversity of living nature. For now, evolution is a wonderful mystery, ripe for further scientific exploration. The gates have been blown open once again. And as a science fiction writer, I'd like to make two provocative and possibly ridiculous predictions. The first is that the more viruses may be found in an organism and its genome, the more rapid will be that organism's rate of mutation and evolution. And the second, bacteria are such wonderful slimmed-down organisms, lacking introns and all the persiflage of eukaryotic biology. It seems to me that rather than bacteria being primitive and the nucleated cells evolved from them, the reverse could be true. Bacteria may once have occupied large, primitive eukaryotic cells, perhaps similar to those seen in the fossil endobionts. There, they evolved and swam within the relative safety of the membranous sacs, providing various services, including respiration. They may have eventually left these sacs and become both wandering minstrels and predators, serving and or attacking other sacs in the primitive seas. Eventually, as these eukaryotic cells advanced, and perhaps as the result of a particularly vicious cycle of bacterial predation, they shed nearly all their bacterial hangers-on in a protracted phase of mutual separation, lasting hundreds of millions or even billions of years. And that what the now trim and super efficient bacteria, the sports cars of modern biology, left behind were the most slavish and servile members of that former internal community, the mitochondria. Which group will prove to have made the right decision, to have taken the longest and the most lasting road? I don't know.